Thank you, Chris, for that broad introduction. Really appreciate the time you spent. Before I start, I really want to thank you for kickstarting and sharing today. And also my sincere thanks to Ahmed Osman for inviting me to talk at this meeting. It's really a privilege to work with your team, Ahmed, and you guys are rain or shine. You're bringing this meeting every year. So thanks again for bringing the community together in this new normal. Today I'm going to talk about, what are some of the emerging opportunities? We have been talking about drug delivery for quite some time now. In the last, particularly since the, drug delivery made a big comeback from 2010 onwards or so. There was a big push for drug delivery-based products, particularly some of the work led by ALZA and J&J on transdermal and other areas, but there's a big push that came in the 2010. So today I'm going to talk about, are we at another inflection point where we're going to look at some new barriers, new modalities, or are we going to continue the kind of things that we were doing before? Of course, we need to have both in a happy medium. So please feel free to send me your questions, and there'll also be time at the end. So having said that, I'll briefly talk about some of the ongoing work that all of us are doing. And I would really focus today's keynote on the emerging trends, some of the new areas, the new frontiers that all of us can look to work, and along the lines talk about future directions and opportunities. So this is something that, if you've gone to any conference in the last 10 years or so, these are some of the big areas that we have heard, and we have made some good progress on each of these topics, right? So drug delivery has really made broad utilities. Be it oral delivery of peptide. We have seen so much research work on delivering peptides orally versus an injection, but now we have an approved product from Novo on oral GLP-1. So clearly, drug delivery has enabled that people to move away from injections. And Chris talked about patient centricity. That's a huge patient-centric activity, right? And then also, we all heard about how folks have been working on high concentrated biologics, because there has been a big drive to move high volume IV infusions to a high concentrated sub-Q or through a wearable device, which is my third point here. So folks have been working on how can we really make high concentrated biologics through, whether it be protein engineering, whether it's be using crystallization, spray drying, or simply by other means like the age-old lyophilization. There are multiple technologies that we have heard about how drug delivery has played a role there. Along the same lines, reducing the frequency of injections, folks have been working on sustained release of protein. So how can we bring people less frequent into the hospital setting by having a large volume of dose released over a longer period of time, including half-life extension. Naturally, oral biologics have been an area that many technology companies have been looking into, and many organizations such as ours, but we haven't quite there yet in terms of an oral product on biologics, unlike the peptides. And of course, as we know, 40% of the drugs fail, oral drugs fail in drug development, and they need solubility improvement. So again, this may not be a complete list. I haven't talked about other routes of administration, such as, transdermal or inhalation, but clearly you get the message, right? We have been really moving away from traditional areas, whether it's transdermal or inhalation, to some of these novel aspects. And I called out a few of them here, but essentially that's what all of us have been really working on, and we have made tremendous progress. So it's great to be part of such a community, and where the drug delivery and formulation has really brought upon improved aspects in human life. So now we are at a point where that is going on really well. We have a number of scientific expertise happening in those areas that I talked about. But what else? What is going to be our next big thing in drug delivery? So even though these are areas that people have been working on, I feel that the momentum has come about right now, especially in the last year and a half to two, where some of these areas that I'm going to be talking about have new momentum has been established. So if you take, the RNA delivery, so whether it is gene therapy, whether you need to be delivering RNA, whether it's mRNA or DNA, or different forms of the RNA, whether it's siRNA or saRNA or shRNA for that matter, we have been really looking to how do we really use the RNA technologies to deliver to patients so that we can create the biologics which is required for the patient instead of actually delivering the biologics. So the proteins will be created by the body. So this is an exciting area, and you'll see more when I talk about how this is going to have an impact. And then the other area that is really emerging is cell therapy, right? As you know, cell therapy has been a product now, especially in some areas. We have seen people getting better and cured. Now, how can we take cell therapy to the next level? Is there a way to scale cell therapy so that it can be affordable and also delivered in a patient-centric manner? So that's an area that's really booming. And gene therapy in terms of delivering virus, the adeno-associated virus, AAV, has been phenomenally making a lot of news. As you know, there are approved products in the area of AAVs. So we'll look at AAV as a subtopic today. And the last two are also very exciting for me. These are not in any order. But if you look at number 4 I have in the list is immune targeting nanoparticles. So this is an exciting area. You may have seen me talk about this in other forums, but what I want to mention is, traditionally we have used nanomedicine for nanoparticle technologies for either solubility improvement, or we were trying to do some controlled release and also some passive targeting based on EPR effect or controlled release for that matter. You've seen nanomedicines advancing. The immune targeted nanoparticles is slightly different in that the nanoparticles are targeting the immune system versus it's actually targeting the cells, it's targeting the tissues or the organs. So previously we have been looking at how can we get nanoparticles into the tumor site, tumor tissues. Now we're saying, wait a minute, if we can, the immuno-oncology advances have really taught us that maybe we can target the immune cells and that will do the trick for us. So that's an emerging area that some of us have been working on, and it's quite exciting. And the last one is antibody drug conjugates. And it's phenomenal. In the last 2 years, at least 3 to 4 ADCs have been approved. ADCs have made a significant comeback in the industry. And ADCs are, essentially you have a cytotoxic payload. It doesn't have to be cytotoxic. It can be any payload, small molecule payload, attached to a linker and actively targeted by an antibody. So the antibody conjugates are really making a big push now for a number of areas, not only ADC, but also you can use antibody-based delivery for even the RNAs. So in my opinion, I think these are areas that have clearly shown that they're going to grow in the next coming years. And again, by far, this is not a full, complete list. There could be other topics that which is not in this list, but I just wanted to, given the context of the presentation today, I thought I'll highlight some of these. So in terms of nucleotide-based therapeutics, there are severe delivery challenges, right? So clearly, whether it's the oligonucleotides or the siRNA or mRNA, you really need to be delivered to the process of endocytosis. Because if you're not able to penetrate inside the cell and enable endosomal escape, you don't get the protein generation required. So this is quite a challenging task, and this is why it makes this a great opportunity as well. So there are a couple of steps for the endosomal escape process. In the cytoplasm, so essentially you have the early endosome, you have the late endosome, and the lysosome compartment. So clearly there are multiple barriers for one to go through to enable any of these RNA-based technologies, whether it is single-stranded or double-stranded. So this is an area that, previously we were just delivering to the bloodstream plasma or to the tissues or tumor. Now we have to enable this multi-layer absorption process. So it's clearly overcoming absorption, but within multiple layers. So this is where some of us really need to work on, and all of you, for that matter, to bring new technologies so that we can understand this process really well. Why this is exciting is because we have already shown in the last 2 years is that the nanoparticle-based system, essentially lipid-based nanoparticle, can really make some of this happen. Essentially, we now know that even though it is not for therapeutic applications, LNPs has been an approved product in the area of vaccines, especially in the pandemic time right now. As you know, there are multiple LNP-based formulations available for people across the globe. So we have shown that LNPs can work. Now, how can we shift the focus from a vaccine perspective to a therapeutic class using whether it's LNP or it could be other polymeric particle? But clearly, building on the victory of what we know has worked is something that would be really great for us to build on. So if you think of a nanoparticle platform, you need to have tunability is very important. The payload flexibility, whether it is a DNA, mRNA, siRNA, or small molecule, how do we really incorporate them in the nano system? And also, we need to show that they are stable. They are, you can even conjugate them with an antibody so that we can take it to different types of cells, because as you know, cells can be tagged with antibodies. So if you use similar antibodies, we can be tagging them to the cells of interest. So that's the whole bit of RNA. How can we really use nanomedicines for RNA? I'm trying to switch to cell therapy. So if you have a universal cell platform, whether it is embryonic stem cells or other cells, cell platform usually requires long-term immunosuppression, and the safety and efficacy are really important for the stem cell derived or the transformed cells. So if you look at the figure on the right, what you see is you have the cells encapsulated in an area which is inside the circle, and there is a porous membrane because you need oxygen and nutrients for the cells to survive. So clearly you can't have a medium where the cells are no longer functional. And the cells would then, you need to be protecting the cells from the host immune cells, because anytime you see new cells, host immune cells are going to really go and attack them. So, and that's where the cell, so once you have this little drug delivery encapsulation, you have the proteins developed, and the proteins are now delivered through the, comes out of the pouch. So it is really important to secure them using an encapsulation technology. And the technology need to have a couple of these salient features, such as they have to be immune protective and biocompatible. They have to be retrievable, because in case if you see a side effect, we should be able to fish it out and take it out. And immunosuppression activity and kills, which are two important things required there, because in case if something side effect happens, you should be able to kill the cells. They should be applicable for local as well as chronic systemic delivery, applicability to broad cell types, and vascularization is really important, because otherwise the cells don't really survive, as I mentioned before. So this is a fantastic opportunity. There are not too many off-the-shelf technologies out there which can provide long access to cells. So this is an area that I would say that would be emerging in the coming years. And now moving to AAV, I mentioned this, that AAV is really an exciting area. There are a lot of activities going on, a lot of funding happening. Basically, the RNA-associated virus, they infect and divide the non-dividing cells. They integrate non-integrating virus. The duration of gene expression is dependent on how the turnover of the target cells happen. And there's a structure given there for AAV genome. But one of the key issues for AAV is tropism. Just like nanoparticles would go to the liver, most of them, AAVs have a specific tropism. And if you see in this picture, AAV 1, 6, 7, 8, and 9, they really literally go to the eye, while there are some tropism to the brain, liver, skeletal muscle, and so on. So there are 13 naturally occurring AAV serotypes currently out there, and they really expresses diverse tropism, and they can also vary across species. So this is an interesting area. how do we use our biology to understand and regular technology understand detargeting? Let's say if you're working with a certain AAV, can we have it detarget from other tissues and focus on one of the organs or of interest for us? And can we also really manipulate the way in which immunogenicity might happen? Can we do some technologies to reduce immunogenicity and also have a switch aspect for AAV? So this is an exciting area. As you know, there are a couple of approved products already. So those are some of the reasons to believe in why AAVs are going here to stay. There are 2 products approved in the last 4 years or so from Spark and also Avexis. So clearly there is a lot of promise in this. Of course, we need to understand the tox and how can we really mitigate the tox. But this is a very exciting area for drug delivery. And I want to now go into the last topic, which is the nanotechnology and ADCs. And I think I briefly talked about this. Nano has been around for quite a number of years. Started with nanocrystals for both oral delivery, solubility improvement, from 70s to 90s. And then nano has been also explored for parenteral delivery. There are several products which are parenteral nanomedicine, like doxorubicin, and then they're also being explored for passive tumor targeting. But clearly, some of the research ongoing is essentially active targeted nanoparticles and gene delivery using nanomedicine. And also the topic that I mentioned, can we use nano to target immune cells? So, so in order for nanoparticles to modulate the immune system, there are a couple of handles that we can use. It could be the charge and size of the particles, the protein binding, how is it shaped, and administration routes are important, sub-Q versus intramuscular versus ID, and the composition of and elasticity. So basically, there is a systematic work going on in our labs where we can look at how each of this affects the uptake of immune cells by the nanoparticles, or immune cells uptaking nanoparticles. So essentially, the nanoparticles, as they are injected, they interact with APCs or antigen-presenting cells, and from there on, it goes forward. Again, the reasons to believe for nanoparticle to work is mainly coming from the cancer immunotherapy, where essentially, from a traditional chemotherapy, in the case of IO, the immune-targeted, the immunotherapeutic molecules, they activate the immune cells, and essentially they go and attack the cancer cells. So this is what we are thinking would happen for nanoparticle also. And some of these beliefs have come from a phenomenon called abscopal effect, where essentially you irradiate one tumor and the antigens come out of the tumor, they get absorbed in the antigen-presenting cells, they go to the lymphatic vessel, lymph node, where it unleashes activated T cells, and you get the T cells go and work in your favor to mitigate other tumors. So can we really utilize nanoparticles for this? So this is where the APCs I mentioned, right? So injecting one tumor and you get effect on a tumor which is maybe diametrically opposite to it based on T cell activation. This is where folks have shown that nanoparticles can really absorb the antigens coming from the treated tumor, whether it's irradiated or whether it is injected one way or the other. And they really bring the antigen-presenting cells to the lymph node, where it really unleashes the CD8 plus and T cells, which can go after anywhere in the immune system. So this is an area that we are talking and working within our groups. It's really important because this way, if there is memory effect, this can keep going on. There is no need of frequent dosing, potentially. And also, we are trying to understand, immune suppression pathways that we can also incorporate, because if you don't need too much, you don't want too much immune activation. So can we also build in some immune suppression pathways while we do this? This is an exciting topic. So now going to the ADCs, antibody-drug conjugates that I talked about, which is the last of the emerging areas. I don't want to call this emerging but in the last few years, it has really emerged. If you look at 2017 onwards, you've seen a number of approved ADCs. And essentially, folks the first ADC was approved in 20 years. in 2000. So you can assume that maybe folks have started working 10 to 15 years before that. So we have a span of 30, 35 years of ADC research, if you do the math. And then we have about 10 products approved. So it's pretty good, right? If you look at the average, it's 3.5 years for a product. And in the last few years, particularly, people have understood how to tune the cytotoxicity of the molecule, the potency of the drug, and how can we really use antibodies to enable targeting. So in ADC's mechanism of action, I'm not going to go through this in detail, but you have a payload, and then you have a linker attached to the antibody. So it's a ternary system. You can work on the PI and other aspects of the antibody. You can look at the hydrophilicity, hydrophobicity of the linker, and also you can work on the payload, the log P aspect. So a number of these mechanisms will enable it into the efficacy coming out of ADCs. How do we design next generation of ADCs? ADCs come with a lot of off-target toxicity. This is one of the primary reasons why ADCs have some issues. This has been a major hurdle for the advancement of ADC therapeutics. So this is where a lot of focus has been going on. How do we really use the antibody to our advantage? How do we conjugate the antibodies to the drugs using whether it's site-specific conjugation or non-specific conjugation? How do we increase the antibody-drug ratio? Because sometimes they're limited by 1-to-1, one antibody to one drug. Can we use increase the DAR value? And can we play with the hydrophobicity and hydrophilicity of the linker so that the entire unit functions as one moiety? How are the PK properties of the antibodies affecting the overall ADC format? So these are some of the areas that a lot of work has been going on. Somehow the traditional drug delivery community, in my opinion, hasn't looked at ADCs so much. They have been looking into so many other areas, but this is an area where a lot of chemistry and drug delivery experience can be put into use. So I'm going to conclude this and leave some time for questions. So in summary, clearly RNA, cell therapy, and AAV are future modalities that you're going to be seeing a lot in the coming years for widespread disease management. Nanoparticles can be moved a notch up higher by looking at immune targeting nanoparticles, which are more amenable and reproducible than looking at tumor targeting because of this liver tropism by nanoparticles, the reproducibility, things like that. And then targeted delivery, using antibodies or whether it is using antibodies to deliver RNA or nanomedicine, that's going to be a key enabler, including ADCs. So that's kind of some of the things that we can bet on as we go forward. In terms of new modalities, basically our vision is to really deliver long-lasting therapeutics. We're using these new modalities. It could be gene to patient, delivering a gene to patient so that it'll really make the biologics, the antibodies. It could be a vaccine platform, say intracellular targeting. You can deliver a virus to patient. You can deliver, that's many of the AAV technologies that you've seen. And you can also deliver cells to patient using cell therapy and next generation ADCs. So I really feel that these four broad classes are what's going to be dictating the next higher end of drug delivery. Lastly, it's a question of, what do you choose, right? You have some of these emerging options. How do we choose between, should I be looking at gene delivery or, whether it's going to be an AAV or cell or simply ADC? So we need to really understand the disease. That's the primary importance, right? Understand the disease aspect, what brings efficacy and safety for patients, and are there any patient centricity needs that needs to be decided before understanding the modality? And then whether it's a single dose or whether it's going to be multiple doses is another big question. And then once you understand the disease aspect, we can decide the modality, what would serve best. Sometimes you may need to bet on more than one thing. And then really build the technology platform. Build the technology so that it can improve the targeting aspects, it can reduce the toxicity and improve manufacturability. and then create platform technology so that you can plug and play when you need when your pipeline advances, and quick transition to clinic, lower cost of goods. So the number of aspects are tuned together. So I hope I was able to give you an overview of what some of the emerging drug delivery technologies are going to be and areas are going to be. With that, I want to summarize my talk. I want to thank a number of colleagues from AstraZeneca, Legacy MedImmune, and some of the other groups and collaborators that I work with. The list is really long. That's why I didn't put an acknowledgement slide, but on behalf of all my colleagues at AstraZeneca, different groups within AstraZeneca, and our drug delivery cohesive teams, I want to thank the organizers once again, and really appreciate your attention and happy to answer any questions. Thank you.